last of the environmental factors, nutrients. Okay, so I'm going to talk about nutrients. Like every chapter, the first one is more about the basics, the second one is more applied. Okay, in the basics section, I'm going to talk about different nutrients, macro and micronutrients, how they affect plant growth, and how do you identify their deficiencies. Okay, the later part, which will happen next week, not this week because I'm traveling tomorrow. The next week when I come back, I'm going to talk about EC and pH, electrical conductivity and pH. Very, very important concepts in greenhouse crop production. Okay, but let's start with micro and macronutrients, or macro and micronutrients, and their effects on plant growth. Who knows the different nutrients plants need? Can you start naming them? I'll count them. Okay, so let's go. So she said carbon, hydrogen, oxygen. Do you apply carbon, hydrogen, oxygen through fertilizer? So how, naturally how? What process? What is the process through which they get carbon, hydrogen, oxygen? Yes. You don't need to apply them. Plants photosynthesize and they fix carbon. And of course, they are structural compounds and they comprise almost 85 to 90%. I get it. But we are talking about nutrients, fertilizer nutrients. Give me the names of the nutrients. One at a time. Nitrogen, boron, sulfur, zinc. We're getting all of them, isn't it? Magnesium, a few nickel, chlorine, copper. These are kind of small things, right? So that is my next slide. By the way, you're already there. Okay, look at this. Before we get there, what I'm talking about is something called essential elements. So if you take a plant and burn it, make it ash, pretty much ash, nothing, plant become ash, and then you take that ash and run through chemical analysis to see what elements are there in that ash. First of all, you won't find carbon, hydrogen, and oxygen because they escape as carbon dioxide, water vapor, and oxygen is part of that. But you find every other inorganic elements. So those three are called organic elements. Carbon, hydrogen, oxygen are organic elements. The inorganic elements, you'll find a lot of them. In fact, you'll find more than these. There are all kinds of things. You find silicon. In some cases, you find all kinds of polluted elements in the plant. They are not all. It doesn't mean that if the element is there in the plant ash, it doesn't mean that plants require them. Simply plants got them because they were in the soil. Then came the question, what do really plants need? There are all kinds of elements, but out of them, what are the elements that plants need? And those are called essential elements. The ones that plants need are called essential elements, and the definition for them is, in the absence of these essential elements, a plant cannot complete life cycle. The number two criterion is, the function of the element must not be replaced by another element. For example, if nitrogen is an essential element, the function of nitrogen cannot be replaced by phosphorus. In that case, nitrogen is not essential. And the third one is, the element must be directly involved in plant metabolism. If these three criteria are met, or at least one of these criterion is met, then it is called an essential element. And when they looked at these three criteria, they found out 14 are essential. And those 14 are shown here. Most of them you already said. What I'm doing here is I'm categorizing them into three groups: primary macro, secondary macro, and micronutrients. Nitrogen, phosphorus, potassium, NPK, calcium, magnesium, sulfur as secondary macros, and then becomes all micros: iron, manganese, molybdenum, boron, copper, zinc, nickel, chlorine. Okay, 14, 8 plus 3 plus 3. The word micro does not mean, very, very important, the word micro does not mean they are less important. They are just required in small quantities. Their importance is as same as anything else, but it's just that their content, their presence, their amounts in the tissue is small, so their requirement is small. That's all it is. Okay. All right. So this slide tells you the percentages of those 14 or so elements. Okay. Again, like I said, carbon, hydrogen, oxygen, the three organic elements, They occupy almost 85 to 90% of the plant by dry mass. The remaining are these. So you can see here, you take a tiny part of that plant, make it into an ash and run through analysis. This is what you see. Nitrogen is 4.5%. Phosphorus is just 0.5% in the plant. Potassium is also 5%, and you can see calcium, magnesium, sulfur about 0.5. Sorry, potassium is 5%, calcium, magnesium, sulfur 0.5, 0.3, 0.3 each. But then when you get to micros, their numbers are so small, all the way to trace values. This was the starting point to develop fertilizer guidelines, by the way. We now know that this is the amount of nutrients that are there in the plant. Then, as long as the fertilizer supplies these percentages in the tissue, then the plant is happy. This information is so crucial. So almost you can see now most of the fertilizers now, if you see 20, 10, 20, this and that, 15, 5, 15, all those fertilizers, you will see N and K are more simply because we know that their percentages are high. You can do the math. 5 plus 4 is 9.5%. This is 10%. This is about another 1%, 11%. All of this put together, not even half percent. 11.5% is all here. But what about the rest 89 or 88.5%? That is CHO, carbon, hydrogen, oxygen. Okay. The three major ones, carbon, hydrogen, oxygen, are from air. We don't supply them. Plants fix them through photosynthesis. But these are the essential elements that we supply, inorganic ones, through fertilizer, through soil. Okay. All right, let's talk about nitrogen. We're going to look at different elements and kind of understand why they are important, what they do. Okay. Nitrogen is required for the vegetative growth of plants. Very important. Okay. Plants become stunted, they become small because of reduced photosynthesis. And also, when the nitrogen deficiency is there, the leaves become yellow in color. Look at here. This plant was grown with nitrogen sufficiency. This plant was grown with nitrogen deficiency. What are the two major differences? What is the number one difference? What is the number one difference? Stunted growth. Less photosynthesis, stunted growth. Number two, what do you see? Where are the yellow leaves? Yeah, because nitrogen is mobile. It moves inside the plant, and if there is deficiency, it moves from older leaves to younger leaves. Why is it moving to younger leaves? No, why is it moving? That's where everything is happening at the top, young leaves. So the plants are smart. They move the nitrogen. If it is deficient, they remove the old leaves and they move the nitrogen up to the top where most of the photosynthesis is happening. So obviously, if that happens, where do you see the deficient symptoms? In the older leaves. Okay. This is another characteristic morphological thing that you will see with nitrogen deficiency. You see here, as you increase nitrogen supply, watch what's happening to root growth. When the nitrogen is deficient, when the nitrogen is sufficient, what's the big difference with root growth?
The root is going sideways and not bigger.
When the nitrogen is deficient, plants produce much more deeper, big root systems. Why? Why do they need big root systems? They are scanning the ground. They are looking for nitrogen. It's deficient, so let's go to areas wherever the nitrogen is, then start taking it. So they have to produce big root system. But you supply a lot of nitrogen, they don't need to spend any resources on the roots. There's plenty of nitrogen. They put everything on the shoot system. This term, this partitioning of carbon, sugars, photosynthase between shoot and root, there is a term for that we use in calculation, what's called root to shoot ratio or root weight ratio, whatever that is called. In general, root to shoot ratio decreases as you give more nitrogen. Root to shoot ratio increases when you give less nitrogen, meaning roots become a larger proportion. The same thing may happen also with the drought stress and nitrogen stress. Both cases, plants are scanning for water or for nutrients, so they have to put bigger roots. But where is the carbon coming from for the bigger roots? By reducing the shoot growth, they take the carbon from the shoot, from the leaves, put it in the root system. Simple partitioning changes. Okay? Please. Sure. We just started. Maybe around half an hour more. 11:15. We'll finish it up. Thank you. What was that? A little bit more about nitrogen. Some facts about nitrogen. You see interesting facts. Nitrogen is there in the air, right? A lot of nitrogen is there in the air. For example, if you take 1 acre of land, somebody calculated this, there's 75 million pounds of nitrogen is there in the air. A lot. But not even a single pound is available to plants because that nitrogen gas is no use. There's a ton of nitrogen, but not useful. Okay, remember that. But plants are smart. They can take that and fix it biologically. You know that biological nitrogen fixation, rhizobium bacterium, or it can be converted. So basically, nitrogen gets converted to ammonical form, and from there it is either assimilated by the plants, or it can be converted to nitrate by nitrosomonas or nitrobacter. There is a chemical process, what we call Haber-Bosch process. This was actually discovered in the Second World War. They wanted to prepare actually, ammunition bombs. There was an accidental discovery. In Germany, they discovered this Haber-Bosch process where they actually used pressure to take that nitrogen in the air, combine with hydrogen under a huge pressure to create nitric acid. And this ammonia basically, and then from there nitric acid. And this nitric acid is used in fertilizers right now, nowadays. But that process was discovered in 1945. Highly energy inefficient process. We all use that. In all countries use this. It's the only process available to get that atmospheric nitrogen, combine with hydrogen, make ammonia, add oxygen and water to that ammonia, prepare nitric acid. Then nitric acid can be used as a fertilizer. That is the only chemical process that is out there. Otherwise, plants rely on their own biological fixation. Those are the two ways nitrogen is fixed or manufactured. The other thing I want to talk about is nitrogen exists in, I know it's a nitrogen fertilizer, but there are 3 forms of fertilizer, 3 separate forms. Urea is one form, CONH2 twice. Ammonium form is there, NH4 plus, ammoniacal. Ammonia is different, ammoniacal is different. Ammonia is NH3. Ammoniacal form is an ion, NH4 plus, okay? And then nitrate form is there, NO3 minus. These are the 3 forms of nitrogen. See, here is an example of fertilizer. Total nitrogen is what? How much percentage? 20%. Out of the 20%, ammoniacal nitrogen is 8%, nitrate nitrogen is 12%. So this is how the normally greenhouse fertilizers are. You'll get at least 2 or sometimes 3 forms of nitrogen in it. And the form you choose will affect the pH. And we'll talk about that next week. But I want you to know that nitrogen comes in 2 or 3 forms in the fertilizer. And you need to know that, very important. Okay? And by the way, before we go there, you can see that fertilizer also has many other elements we talked about, isn't it? Not all of them, but most of them. So you may have to combine 2 different fertilizers to get all those 14 elements. Okay? This is water soluble fertilizer. You take it and you mix it in water. You prepare your fertilizer solution and you run it through plants. Alright, so let's look at the dynamics of all those 3 forms. You can apply urea, you can apply ammoniacal form, but both urea and ammoniacal form can be directly taken by the roots, or urea gets converted into ammoniacal form in the soil with the action of bacteria, and from ammoniacal form it can get converted to nitrate as using nitrosomonas and warm temperature. So these are all interconversions. The nitrate can be taken by the plant. So whatever you apply, urea form, nitrate form, ammoniacal form, these are all interconversions. Urea can get converted into ammoniacal form can get into nitrate form. Urea can be directly taken by the roots, ammoniacal form can also be directly taken by the roots, and nitrate can also be directly taken by the roots. These are all interconversions that happen. Okay? But when you apply nitrate, nitrate is taken by the roots, then what happens is the nitrate goes to the leaf, where it uses the energy from photosynthesis process and gets converted to ammonical form. So eventually, even nitrate is becoming what? Ammonical form. The reason is nitrogen is the key component of proteins, amino acids, and NH2 amide form is the key component in all these proteins. Therefore, all of these nitrates, urea, everything has to get converted to an ammonical form, and that ammonical form is now assimilated into amino acids and proteins, and that's what plants use for growth. Okay, Rubisco, an enzyme used in photosynthesis, 50% of Rubisco enzyme is nitrogen. Is it nitrate form? No, it is in that amide form. Okay, but these are interconversions. Whatever you apply, end of the day, that's what happens up here in the leaves. All right, let's look at compare them, two of them, nitrate form, ammonium form. Then you may ask me this question, which form should I apply, right? Should I just buy a fertilizer with a lot of nitrates in it, or should I buy a fertilizer with a lot of ammonical form in it, right? There are pros and cons for that too. Okay, let's look at the pros and advantages and disadvantages of nitrate, and then we'll get to the ammonium form. Nitrates are easily soluble. As soon as you apply in water, they get soluble, so plants can easily take them. They don't compete with calcium, magnesium, and potassium, whereas ammonical form can compete. Why? They're all cations. Ammonical form, calcium, magnesium, iron, they're all cations. So they are kind of competing for the sites on the substrate particles. Remember, I told you cation exchange capacity. The substrate is negatively charged, and all these ions can get absorbed on the surface, but the spaces is limited, and all of these are competing for those spaces, right? So that's the reason why this form is negatively charged. It won't compete, but it is also not being held. It can get leached because this is an anion, and substrates can't hold anions well. There is some study that shows that when you apply nitrates, plants get little bit tolerant to stress. Little bit not proven, maybe research for one of you. You can compare stress tolerance in ammonical, when you apply ammonical versus nitrate forms. Basically, I grow plants with ammonical form, basically grow with the nitrate form of nitrogen and expose them to stress and see which one tolerates stress better. People are saying that there is some evidence that nitrate, when you supply nitrate form of nitrogen, plants get little bit tough. They tolerate stress. But what are the disadvantages? You know, leaching losses. Why? It's not absorbed by the clays or substrate particles. It's always in the solution, so it can get leached out, wasted. Nitrates, so there is something called, in the plants, you know, plants are like some of us who don't pay attention to what they eat, right? Some of us, we want to eat like that. Plants are somewhat like that when it comes to nitrates. The term for that is called luxury nitrogen consumption. Okay, when you give a lot of nitrogen, plants simply take all of it. They're not going to say, I only need this much, I don't need that. No, they won't do that. They take all of it. They take all of it, and then they store that nitrate, if it is especially nitrate form, in vacuoles of the cells. You see, a cell, the big component of the cell is a vacuole. In a mature cell, vacuole is 90%. Everything else is along the margins, right? All of the nitrates get stored in that vacuoles. They are simply consuming and storing, consuming and storing. The reason why plants do that is they are preparing for a situation when nitrogen becomes deficient, use that nitrate, that stored nitrate. Plants are only being optimistic. But there's a problem with that, especially with leafy greens, spinach, pollock. Is pollock spinach? Yeah, pollock, or lettuce. What happens is when you apply nitrogen, these plants are taking and taking and taking nitrates, storing in the vacuoles, and then we eat them fresh, sometimes in salads. All of those nitrates, where are they going now? Into our stomach. See, the number one cause of nitrate pollution is water, irrigation water, drinking water, I get it. But the number two cause of nitrate pollution for us is leafy greens. And the reason for that is this: nitrates can get stored in the vacuoles. So in countries, in European countries where there are a lot more stringent regulations and what farmers can grow and what farmers can sell to consumers, they actually check the nitrate levels in the leafy greens before, and there is a cutoff. If the nitrate levels are higher than that, the farmer cannot sell to the consumers. When you go to the West United States, more so in Europe, Europe is really, really stringent. European laws are so tight, they're all consumer focused. They want to make sure that it's safe, they don't like GMOs, all of that stuff. But there, you will see this kind of good regulation. And these are good stuff, this is good stuff. They're only trying to protect our health, right? One day, I hope in India also something like that happens, but we don't know what we eat, right? Here. But then let's keep moving on. Again, I showed you, when you take nitrate, the nitrate goes to the leaves, right? Where it uses photosynthesis, energy from photosynthesis, then converts to amide form. So it requires some energy. So that's kind of like an extra step where it might take some energy. And for that reason, actually, when you supply plants with, DAP, diammonium phosphate, it's an ammonical form, right? If you supply DAP to plants, and if you're a farmer, you will notice that the leaves are really dark green with DAP compared with calcium nitrate or something like that. The reason is basically that directly the amide form can be used in preparing proteins. Chlorophyll is a protein, right? End of the day, it's a pigment though, but it's got a lot of nitrogen in it, and it can simply assimilate the nitrogen from amide form. So it gets really, really dark foliage when you apply diammonium phosphate compared to other forms. But there is another concern here, micronutrient deficiency to pH imbalance, and I have not talked about this, which I probably will talk in the pH class. But what happens is when plants take nitrate is an anion, negatively charged anion. So if plants take more of this negatively charged anions, they have to do some pH balance inside the cells. For that, what they do is they send out H plus ions out into the substrate. So think of a root. The root is absorbing nitrates. The nitrates are going into the root. But let's say a lot of the negatively charged nitrate goes into the root. Inside the cells, it's a lot more negative charges now. So they want to do ionic balance inside the cells. For that, you know what they do? They send out positively charged ions like H plus, hydrogen. When there is a lot of H plus in the soil, what is pH? Negative logarithm of hydrogen ion concentration. So the pH goes down and the substrate becomes acidic. See, here is a reason why I told you pH is very dynamic. It changes by crop. And I'm telling you, it also changes by fertilizer that you supply. Because plants want to make ionic balance, they simply send out more H plus out, and when there is more H plus outside, that's less pH, more acidic nature. Okay? Let's look at ammonium form. Ammonium form is good advantage, it's directly incorporated into amino acids. So it's very easy, no extra steps. For nitrate, it has to go up to the leaves, take the energy, then become amide, and then become incorporated. But here is a direct step, so less effort on the plants. Results in large foliage and lush green plants. This is the same reason, large foliage. It's not easily leached in soil as substrates as nitrates. Why? Yeah, it's a cation. It's a cation. But there are disadvantages, especially in hydroponics. If you are growing crops in hydroponics with pure liquid culture and you apply a fertilizer which has got a lot of ammonical form, in hydroponic systems that ammonical form escapes as a gas, ammonia gas. Okay? That's the reason why we recommend nitrate form of fertilizer in hydroponics than an ammonical form of nitrogen, because it can escape as as ammonia, especially when pH is high. That happens when pH is high. Can compete with other positively charged ions and can result in blossom end rot. So hold on to that blossom end rot. I'm going to show you some examples, but it's calcium deficiency. Okay. Calcium is also a cation. Ammonium is also a cation. They compete for each other, and so being ammonium NH4 plus, it's a bigger one. It can occupy more space, and then calcium deficiency results because of that. Calcium deficiency is happening because calcium is not being absorbed by the substrate. Ammonium is competing for those sites. And excess ammonium can cause phytotoxicity. Okay. So which form is better, right? So you got a nitrate here, you got ammonium here. It depends on the situation, it depends on the crop. I will say use nitrates for hydroponic crop production, maybe use ammonical. For people claim it's not valid. People claim that if it is you are growing crops in a place where there's a lot of cloudiness during rainy season, things like that, maybe it's better to use ammonical form than nitrate form, because there is going to be less photosynthesis. Therefore, plants require nitrate form has to go to the leaf and use the photosynthetic energy. It's all competition. Just why not use ammonical form? But it's yet not valid. Some people disagree with that. So it is your choice. Again, you need to know what are the advantages and disadvantages. It comes in two forms again: nitrogen, ammonical, and nitrate form, and these are the advantages and disadvantages of that. All right, let's move on to phosphorus. Um, so phosphorus, what is this? DNA, right? And this is the helix, right? That helix is made up of sugar phosphates. So right there is phosphorus. Phosphorus deficiency can cause what? Basically, it's a structural element of nucleic acids, DNA and RNA, and absolutely required. Adenosine triphosphate, ATP, very, very important energy currency for plants. That's phosphorus. So that's why plants require phosphorus for energy. Phosphorus is all energy. More phosphorus, more energy in the plants, right? Phosphorus also causes decreases in shoot growth and also flower initiation. But more importantly, see phosphorus deficiency, purple margins. How many of you remember sodium iodide tests for starch in your schools? Earlier schools you may have done it, right? You get purplish color. So what happens when phosphorus becomes deficient is the starch gets accumulated. Starch should get converted into glucose and then to sucrose and should get loaded into phloem so it can go to different parts of the leaf. But under phosphorus deficiency, the starch accumulates. It doesn't get converted to glucose and sucrose. That accumulated starch is now showing us that purple color. So classic phosphorus deficiency is purplish margins of the leaves. Okay. We'll move a little faster, but I want to talk one thing about phosphorus, especially if you are in producing seedlings. The recommendation is that don't give seedlings too much phosphorus. Don't give too much phosphorus. I told you phosphorus is all about energy. You give phosphorus, the seedlings will grow big, and that is not good. Okay. See here, there are two types of fertilizers. There is this fertilizer for mature plants, and there is a fertilizer for seedlings. What is the difference? Big difference. Look at the composition. Look at the different fertilizer elements in there. What do you find there?
So in the mature plants, more nitrogen is required than the seedlings.
No, keep going down. What am I talking about? I'm not talking about nitrogen.
Phosphate levels are also low in seedlings.
2%, isn't it? Available phosphate is 2% here for seedlings, whereas here 8%. We want to give a fertilizer which has got low amount of phosphorus for seedlings, especially in floriculture crop production. Because when you give high amount of fertilizer, high amount of phosphorus in the fertilizer, and if the temperatures are warm, those seedlings are going to get lanky, tall, poor quality. It's very difficult to transplant, very difficult to establish them. So we always want to give a fertilizer with less amount of phosphorus, simply because of ATP energy. If you give more phosphorus, more ATP, right? Potassium, next potassium. Potassium activates starch synthase. It's a very, very important enzyme. Starch, synthesis of starch. It's a fundamental thing in metabolism, in carbohydrate metabolism in plants. That is activated by potassium. But also potassium is involved in peptide synthesis. Remember this, these are ribosomes, they're bringing in, transfer RNA is bringing in, and then we are preparing peptides here. That's what this is, messenger RNA. This is all involved in that peptide synthesis. But for a plant physiologist, potassium is absolutely required for stomatal regulation. Opening and closing of stomata happens because of efflux of potassium into and out of guard cells. So very, very important, okay? Potassium deficiency, classic symptom. This is a tomato plant, and if you see margins becoming brown, necrotic, that's a classic potassium deficiency. Okay. Then comes calcium. Calcium, we take calcium, right, for our bones and things like that. It's just like that for even for plants. The equivalent of bones for plants is cell wall, right? That's what it is. So the calcium is an integral part of the calcium pectate, the cell wall structure. So if you don't have proper amount of calcium, that wall structure is not formed, so cells lose their integrity, and basically the tissue dies. And this happens especially in young tissue. See here, calcium deficiency, this is called blossom and rot of tomato. If you see the fruit, at the bottom of the fruit, you see something like that. That young part of the fruit, it should be growing. Those cells are dead simply because there is no calcium pectate in the walls. And that's blossom and rot. This is called tip burn of lettuce. Again, young leaves are becoming necrotic. They are dying simply because there isn't enough calcium. The problem with calcium is calcium does not flow well in the phloem. It has to go through water, through xylem. And so when you get to the fruit or growing tips, the xylem connections, it's not there much. So calcium cannot adequately move into the growing parts. But on the other hand, if there is a warm temperature and plants are expanding rapidly and the calcium is not getting into the young tissue, that causes the death. Rapid growth of the plant with a less amount of calcium going through that young tissue, then it causes necrosis. Tip burn in lettuce, blossom and rot, they both are classic examples, and the death is happening because the cell walls are lacking calcium. They are leaking, they are dying. There is no integrity of the cells. Okay. Calcium also acts like a messenger, a signal. How do plants know that it is cold outside, it is drought, or it's some other external environment stimuli is there? Calcium is one of those. So calcium, something else senses a stress outside, that sends a signal to calcium. Calcium takes the signal to the nucleus, provides the information to the nucleus, and then DNA replication happens, DNA changes, and the new genes are expressed. Gene expression results in some tolerance. So as a messenger, it is carrying the information from the cytoplasm all the way to the nucleus. This is a secondary messenger. Calcium is what is used there. Okay. All right, remember this: tomato blossom and rot and tip burn in lettuce. Again, the young tissue is dying. That is, the young tissue is dying because it is growing and it is not being supplied with calcium. Calcium has to move through xylem, not through phloem. Calcium moves through xylem to get to this young tissue, and the xylem connections are probably not there much in that growing areas. So they won't get calcium enough, especially the tissue is rapidly expanding. The calcium is not being supplied, then it dies simply because calcium pectate is not being formed in the cells. Magnesium. Magnesium is integral part of what? Chlorophyll. Central molecule. This is chlorophyll. Right at the center, what do you have? Magnesium. And what else you have? Nitrogen. But magnesium is also there in the central molecule. So when you have magnesium deficiency, you see intervenal chlorosis. Intervenal, between veins. Intervenal means between veins. Chlorosis, yellowing. Between veins you see yellowing, but the veins are green in color. Remember this deficiency. Intervenal chlorosis. Between veins you see the leaf lamina dying, but the veins remain green in color. Okay. You're going to do all this afternoon in tutorials. Okay. You'll spend a lot of time identifying nutrient deficiencies. I'm going to get you a key. I want them to print it for you, but you're going to spend a lot of time today nutrient deficiencies. But try to understand chlorosis. I said chlorosis, right? Chlorosis means what? Chlorophyll dying. Therefore, when chlorophyll dies, that becomes yellow in color. I said necrosis. Necrosis is basically death of the cells. Brown in color, black in color. Intervenal chlorosis. Between veins, it's chlorosis, but vein itself is not. Vein is green in color. You see the green color veins? But in between, it's turning yellow. That's magnesium deficiency. Okay? Magnesium is integral part of photosynthesis because of chlorophyll. Magnesium also helps in some activation of enzymes like pep carboxylase, glutathione synthase, things like that. But I want you to remember this symptom, deficiency symptom. That's because it's an integral part of chlorophyll. Sulfur. Sulfur is also part of the chlorophyll. It also affects chlorophyll synthesis. And when there is sulfur deficiency, what is this? Everything is looking yellow, isn't it? uniform yellowing of the leaf is sulfur deficiency. You see here, completely sulfur deficiency. All right. So remember that. Magnesium, intervenal chlorosis. Sulfur, complete. But how do you compare this with nitrogen? Nitrogen also chlorosis, complete leaf, but those leaves are at the bottom. But his entire plant is showing here. Okay, those are small tips that you can use to distinguish between what is causing what. This is the micronutrients, and I'm going to keep micronutrients small, but the most important micronutrient iron. But there are also other micronutrients like manganese is essential for enzymes, iron for chlorophyll, molybdenum is required for nitrogen fixation in in biological nitrogen fixation, copper for proteins, and boron, translocation of sugars. All these micronutrients do play a variety of things in metabolism. They're all important. They're just required in small quantities. They're absolutely required for metabolism. Iron is an integral part of chlorophyll. It is required for chlorophyll biosynthesis, and when you have iron deficiency, here you can see what is this called? Is this chlorosis? What is that symptom? It is intervenal chlorosis. I can make this picture bigger if I can, but the leaves are intervenal chlorosis. But here is intervenal chlorosis, and yeah, why lower leaves for iron? But if you go to I'm sorry, give me a second. I'm going to go to magnesium. Remember I showed you intervenal chlorosis for magnesium? These, you cannot able to see this, but these are upper leaves. Okay, but let's go to iron one more time. Older leaves. Why older leaves for iron? Iron is heavy. It cannot move. It's not mobile like nitrogen. I'm sorry. I'm totally wrong here. Iron also moves like nitrogen to the parts where it is required, and it causes the internal chlorosis of the older leaves. Okay, whereas for the magnesium, you will see internal chlorosis of the younger leaves. Okay, but you'll have a key today, this afternoon. I'm going to get you a key, and I'll show you this afternoon after lunch, where you can actually use that key and kind of come up with old leaves, young leaves, death, necrosis, chlorophyll, some things like that, and kind of come down to what element it is. That's the sort of exercise you will do today. I got some examples of pictures of plants showing some deficiencies, and all of you should actually take that key and come down that process and figure out what the deficiency is. Okay, that's what we'll do this afternoon. And in fact, I'm going to ask you to go outside and at least find one nutrient deficiency symptom. Bring it back here with you. It could be an ornamental plant, it could be a weed, it could be any crop. Just bring one and tell what it is. If you bring more, I'm happy too, but let's see what you will find. Okay, I think this is the last one I have. Yeah, this is the last one. So again, let me revise. And this particular chapter, I just introduced you different elements: NPK, calcium, magnesium, sulfur, and all the micros. We talked about nitrogen in depth, why it is important. We also talked about phosphorus, potassium, phosphorus symptoms. We looked at different symptoms, right? Deficiency symptoms. Starch doesn't translocate, so purpling of the margins in phosphorus. Potassium causes necrosis of the margins, right? Why they are important? Potassium is required for stomatal regulations, right? Phosphorus is energy. We want to give less phosphorus to seedlings. We also looked at calcium. Calcium is important for cell wall formation. Calcium pectate. And if cell walls are not formed, there is no integrity of the cell itself. Cells become liquidy. They die. And that's why you see, especially at the tips where there is active cell growth is happening, blossom and rot or tip burn of lettuce, basically that's what it is. And then we looked at sulfur deficiency, we looked at magnesium and iron, we compared intervenal chlorosis, upper and lower leaves, right? So these are all the things, just to get you into what these elements are, why they are important. And then when we talk about EC and pH, we use these concepts and try to talk more applied, how we can use this information to optimize plant growth. Okay, so we'll stop here. I think we have a break. I think you guys can get now, so you can get a break now and get some coffee, tea, whatever they give you, or water, or nothing, whatever that is, and then come back. I don't know. We're meeting at what, 11:15, right? We can do also 11:30. That's fine. Okay.